Hello, everybody. Thank you for joining us on the 20 series of Super Spreader Seminar on the 14th of March, 2024. This session, we had some members attending online and some members are joining via in the lab of the new opened lab of OMNI-RÉUNIS. As always, we start with land acknowledgements. We acknowledge the land of the OMNI-RÉUNIS Network head office, which is located on York University. And York University recognized that many indigenous nations have long-standing relationship with territories upon which York University campus are located that precede the establishment of York University. York University acknowledged its presence on the traditional territory of many indigenous nations. The area known as the Toronto has been caretaken by Anishpac nations, Puruscience, Confederacy, and Huron-Wendat. It's now home to many First Nations, Inuit, and Métis communities. We acknowledge the current treaty holders, the Mississauga of Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Bill Convent, an agreement to peacefully share and care for the Great Lake regions. In the Super Spreader Seminar Series, the OMNI-RÉUNIS Super Spreader Seminar Series welcome faculty members, OMNI-RÉUNIS affiliate, and high qualified persons, including postdoctoral researchers and graduate students, who showcase their research, promote their ideas, share their research experience, and establish connection among various branches of the network. It is a platform to spread scientific knowledge about the One Health research framework and the research progress of over 30 OMNI-RÉUNIS HQPs within the network scientific research community. These seminars facilitate knowledge exchange within OMNI-RÉUNIS, the emerging infectious disease modeling network, and collaborating scientific community. Today we have Shohel Ahmed. Shohel, thank you for accepting our invitation for the 20 series of OMNI-RÉUNIS. Shohel is a PhD student in applied mathematics at the University of Alberta, where he is a key member of interdisciplinary lab for mathematical ecology, and epidemiology. Under the guidance of Dr. Hao Wang, his research delves into the realms of deterministic and stochastic models within mathematical biology, with a particular focus on ecological stoichiometry and the modeling of infectious disease transmission. Currently, his work is centered on exploring how climate factors influence the modeling of the various infectious diseases. As a mathematician with a passion for uncovering the integrated biological pattern in nature, Shohel is dedicated to linking these discoveries to his field of study, aiming to contribute significantly to our understanding of ecological and epidemiological dynamics. Thank you, Shohel. He's going to talk about investigating the climate influence on norovirus spread in the United States. Okay, and let's hear from him. Let me stop sharing and thank you, Shohel, for joining us. Yes, we can see your slide. We have the honor to meet you previously in the lab, OMNI-RÉUNIS lab in the annual meeting. It's great to have you online right now.
Yeah, thank you, Shanaz, for your short and very precise introduction. So today I'm going to talk about the climate influence of the norovirus spread in especially in United States. And I would like to thank OMNI for giving me chance to talk about my current research progress on this seminar series. So mainly my topics is divided into two parts. In the first part, I'm gonna construct the mathematical model related to the spread of the norovirus. And then I'm gonna investigate the power of machine learning to predict the norovirus spread. So let's talk about some motivation behind our study. So basically norovirus is a very vital gastrological disease, which causes around, on an average, almost 20 million infected cases per year in United States. And basic symptoms is basically vomiting, stomach pain, and diarrhea. Norovirus is often called as stomach bug. Sometimes it's known as food poisoning and stomach flu. One of the most common cause symptoms for like the norovirus for the children is like vomiting, and sometimes it goes to diarrhea as well. It's more common for our adults, like for adults is gonna be mostly like diarrhea, as because we have like more immune system. And once the virus reaches the stomach for a long time, it's inflammate, and that inflammate also cause vomiting and stomach pain. So basically, if this is very deadly disease for the children actually, as because like children has less immune system. So that's why almost like 400,000 young children visited emergency room per year in United States due to norovirus infection. And almost like 570 to 800 people die each year due to the norovirus infection. So the problem is actually there is like lots of pathways to transmit this virus. Also, as like you can see the green leaves like food, lettuce, and shellfish, some fresh foods. They are also responsible due to the transmission of the norovirus. If you have a like good health, you can fight with the virus almost like around 1 to 3 days. But like for if you have like less immune system, so it can last long till 4 to 6 days. There is lots of symptom for the norovirus. It depends on your body immune system. Actually, the most common one is basically diarrhea for adults and like for vomiting for children. Other than that, like there is lots of symptoms like stomach pain. Sometimes it can be low-grade fever, dehydration, body acne, chills. All those symptoms are very responsible for the norovirus attack. So just like the other viruses, norovirus is very transmissible. Like why is very transmissible? It can transmit from a close contact with someone, and if you touch any contaminated surface, or if you then touch your mouth or nose, it goes through inside your body and it can transmit in your body. And if you drink any contaminated food or beverage, that's also can cause like for the transmission. So basically, our purpose is to develop infectious disease modeling that can affect through environmental condition, such as temperature and humidity. And we want to introduce different transmission dynamics like direct transmission, indirect transmission in our model to show how norovirus transmitted. So this is like the current situation this is a figure from CDC. So basically we mostly talk about United States as because the data availability. So the during 2012 to 2014, you can see so there's a chunk of infection happens in this like red region. So in that red region between November to April, so there is the infected cases are always higher due to the other month than the other months. So obviously, like that region, we know mostly like winter region. So winter season, so there is more infections. So obviously, there is certain kind of relation between the temperature or like the other environmental condition to the infection case. So as the research goes on, so most of the people find out what's the relation between the temperature and humidity with the norovirus. Back in 2014, one paper is published related to the humidity and temperature. What's the relation between the humidity and temperature to the norovirus in the surface? So if you look at these two columns very closely, you can easily see that with the increase of temperature, the survival rate of the norovirus on the surface is decreasing. And also, humidity is a very real fact. There is mostly like two humidity. You can see relative humidity and like absolute humidity. Even like if you increase the absolute humidity, you can the survival rate of the norovirus goes down. So with this motivation, we have started working on the norovirus, and we found some related work that has been going on, still going on with the prediction and with the relation between the transmission. Back in 2010 and like 2014, the work mostly done on the air temperature and relative humidity, and some works also done in like seasonal persistence and infectivity of norovirus. And in 2017, a study is conducted to investigate the humidity as well as like the human encounters on the transmission of the norovirus. In 2021, there is another work that is done that they showed the infectious dose and the infection of the norovirus can prevail through environment and food. And recent work in 2023, they characterized some of the transmission in with respect to the norovirus genome, and they found that every genome is like very sensitive to the environmental condition. So with with this with this like the previous work in mind, we have set our goal to investigate further, and we started working on a base model that we all know, like SEIR model, susceptible, exposed, infectious, and recovered. So we incorporate some environmental condition like temperature and humidity on the survival of the norovirus on the surface. And then we incorporate some direct and indirect transmission in our model. So we incorporate direct transmission through like 5 compartment. One is susceptible, one is exposed, and symptomatic infected, asymptomatic infected, and recovered individual. And we incorporate indirect transmission through another compartment, like susceptible and infected compartment of foods. So there is lots of foods mostly responsible. So we took shellfish. There is some oyster. Those are also responsible for the norovirus transmission. So let's talk about model construction. So this is the flow diagram of our model construction. So in the first, like there is two half. In the first half, you can see in the top transmission dynamics, there is like the direct transmission. And the red box here is basically represents the compartment of the norovirus in the environment, and the low compartment SF and IF represents the food contaminated compartment. So if I show the differential equations related to that compartmental modeling, so here it represents SH is basically the direct transmission. So this five compartment is responsible for the direct transmission, and P compartment represents the compartment of the norovirus. So in our modeling, we assume that the transmission go through from people to people. That means it can happen through infected, exposed, and asymptotic infectious people as well. And for the norovirus, that's DTH, that the death rate of the virus depends on temperature and humidity. So that death rate of temperature, on using that, we can also see our transmission depends on temperature and humidity as well. For the indirect transmission, we consider susceptible and infected case where alpha P represents the contribution of susceptible to infected case of the norovirus. So coupled with direct and indirect modeling, we use this parameter description of our for our system. And next, what we did in 2020, so they have used survival time for the norovirus on the surface using temperature and relative humidity. We used a linear regression to find out the relation of the survival time of the norovirus on the surface in a function of temperature and humidity. As we know that it's going to degrade if the temperature and humidity increases, so we consider a survival probability function e to the power minus dt to find out the death or mortality rate of the norovirus. And the relation give us that death rate in terms of temperature and humidity by calculating the ln and find out the relation of the death mortality of the norovirus. And we all know that the transmission rate is very vital for any disease transmission. The problem is basically every infectious disease modeling, most of the transmission they consider constant transmission rate, which is not a practical one, as because if you work with like temperature or seasonality, so transmission rate always varies. So back in 2012, my supervisor and his team work on the time-varying transmission rate, which is known as like inverse method, to find out the transmission rate of any infectious disease modeling, especially applied for seasonality. So with that comes with, in a brief, it comes with a three-step process. In the first step, you need to know like weekly infectious data on the ith week. Then you have like a compartment, and you can impose that data inside your compartment for ith week, and individually k is the interval between each time length. And from that step, you can find out the beta, the transmission rate using each compartment from the previous data. And using a long-time data series, you can approximate each time varying transmission rate for beta. You can easily see here we have used DTH, that temperature and humidity dependent deaths in our steps. So obviously the transmission rate we're gonna get that composed with temperature and humidity. So let's show some numerical results. Before that, so this is the study region we have studied. So basically we covered the whole US region that's divided into 4 parts. Demographically is very important, as because if you see like south part is the most like if you compare the temperature and humidity, so south part is very different than the other rest of the part, and northeast part is basically mostly dense due to the population. Midwest is like a little bit cold region, and west region is like the vast for its size. So we covered this 4 region in our study, and same as like the CDC. They also conducted their data collection on the basis of this 4 region. So this is like the average temperature and relative humidity graph for the 4 region. Eventually in the left panel, you see the average temperature, and in the right panel, you see the relative humidity in 4 region in United States. And in the left panel, you can see the green curve has the highest. This is corresponds to the south region of the United States. So obviously the south region is like more heated, and the blue region is basically the Midwest. It's like less temperature, and corresponding to the humidity, also like the green curve represents like less humid, and the blue curve and red curve it corresponds to the northeast part, and the Midwest parts represents most humid region. So if we compare with the death rate of the norovirus, so we compute the death rate of the norovirus in all 4 regions of United States, and you can see the corresponding most mortality rates goes to the south region. Obviously, in the previous slide, I have shown you like south region is the most most hot than the other region, and the blue curve represents like the most transmission, most death, mortality rate, less mortality rate due to its coldness, as well as the west part as well. The peak represents basically the peak seasons is basically the summer seasons. And if you start with from the beginning is basically the winter. So the below region shows the winter mortality rates, and peak is during the summer region. Then we have computed the transmission rate in every region. So starting with the Northeast region, we computed the transmission rate beta T, and we also computed the weekly confirmed cases by fitting notification data, and also the fitted curve from beta. We also obtained using inverse method. We also fitted our curve as well. So starting with the daily confirmed cases, basically in the first seasons is all about all the peaks is during the winter season, and in the below curve, there is all during the summer or like in hot season. So it's seeing that actually like during the winter season, the transmission rate is very high, and also the confirmed case is very high in Northeast region. Same as like the Midwest region, during the summer season, it's goes low in daily confirmed cases, and it goes high during the winter season. We compute the same transmission rate and the confirmed cases for the southern region. It shows same, but like in the early 2023, there is a high peak due to the transmission of the norovirus. And lastly, the Western region shows more high peak in the daily confirmed cases during the early 2023, and it goes down during the summer season as well. Now, if I compare the whole transmission rates and the daily confirmed case of each region, so we can see like the weekly confirmed in terms of weekly confirmed cases, South region goes high during each winter season, and also like the lowest is basically the Northeast region in terms of weekly confirmed cases. But it's in terms of transmission rate, if we compare, like the blue region comes with the Midwest, which is like the coldest region in our study, and it comes with the most transmission rate in on top of like other regions. And on the transmission rate, you can see the green regions, which comes with like most weekly confirmed case, has like less transmission than the others region. Now we started working on the prediction of our outcomes. We use machine learning to find out how we can predict our model to predict the infectious for the next week or like for the next couple of months. So there is like lots of machine learning techniques, but we have used the most popular machine learning techniques, gradient boost machine learning techniques, also known as GBM. We incorporate the transmission rate from the average temperature and relative humidity in our model, and we used a package now known as GBM in our language. You know, like every machine learning technique comes with like two parts. So you need a training data set and you need a testing data set. So we also did the same. We had divided our data set into two parts, testing data set and training data set. And for finding out our results, for the significance of our results, we have also calculated two error. One is mean absolute percentage error, and another one is mean absolute error. And mean percentage error in terms of its compute with respect to the actual value and deviation of the prediction value. And we calculated for each training data set, find out the mean error, MAPE and MAE. So this is the table for our training and testing duration. So we have started with training length of 40 weeks, and we increased by each 4 weeks. And then after the training period, we have test next 2 weeks for the prediction. Like say for example, if we start with like the week 40, if we train our data set for the 40 weeks, then we computed our test duration for the 8 weeks. By this, we increased each 4 weeks, and we also tested our data for the next 8 weeks. So this is the calculated predicted error for our case testing and training. So each training length shows the corresponding MAPE and MAE. Among them, if our training length is 48 days, that give us the less MAPE and MAE. And then we have computed our prediction results for the like next 8 weeks from January 19 to December 3, 2020. We have trained our data for the 48 weeks, and then we predict for the next test our data for the 8 weeks, December 4 to January 28. And we calculated our corresponding fitted results. And we shows that our data, our machine learning can predict pretty well for the next 8 weeks. So for this one, we only use the Western region data. Let's come to the conclusion actually. So basically, we have developed a mathematical model that incorporated through the direct and indirect transmission, and we successfully induced climate dependence effectiveness on the infectious disease transmission. We investigate the influence of environmental factors like temperature and humidity in our model so that we can find out how the atmospheric condition is very important for the disease spread. We applied inverse method to find out time-dependent transmission rates. That's very important for the disease dynamics to show the seasonality. And also we also found the disease transmission varies over time and how climate is very influential. And then we employed machine learning techniques through one of the machine learning a very popular machine learning technique, GVM, to estimate the transmission rate from the average temperature and relative humidity and make prediction. I would like to thank our collaborators, my supervisor Hang Wang, and our postdoc from our lab, Juping. These are the reference I have used during my study. And I would like to thank my University of Alberta, as well as like OMNI, to help me for the funding, and as well as to give me chance to talk and show my research outcomes in this seminar. Thank you.
Thank you, Shohel. Thank you. Thank you for your talk. We just, our heads gets warming about different types of material you presented. I really enjoy it. Thank you very much. If there is any question, please ask. Or yeah, Joseph is raised his hands. Yes, Joseph. Oh, Joseph is clapping. Right. Okay. I just have one question I would like to ask. Do you consider only mortality rate and transmission rate dependent to humidity and temperature?
Yes, like the virus, what I did actually, like the virus depends on temperature and humidity, right? So
Yes.
basically I incorporated the mortality rate of the virus on the surface using temperature and humidity. And that also helps me to find out the transmission rate. So that transmission rate basically depends on that death rate. So that's why like the transmission rate is also like depends on the temperature and humidity.
And one more question. For the time varying transmission, it is somehow Markov change models, right? It depends on the previous stat, final stat.
So basically, in our case, is very straightforward actually. Like it's very like our discretized version actually. Like it's always depends on the other status. Yes. Yeah.
So you change your model to the stochastic one with this time varying uh, time series.
Uh, Not like that actually. Like like say for example, if you have like weekly infected data, so what we
Mhm.
did actually, so for each infected data case, we just calculated the transmission rate in a discrete system, and that discrete system is gonna help me to find out the transmission rate for the next step.
Yeah.
And if you consider like a very small time steps, so then all the time steps gonna be like the transmission rate is gonna be coincide.
Yeah, discrete one. Thank you.
Yeah. Listen.
Thank you very much. Yes, we have one question from Joseph. Yes, Joseph.
Okay, so yeah, thank you, Shohel, for the wonderful talk. So I was thinking about this. So I mean, the virus is influenced by environmental conditions. So you incorporated, I mean, environmental conditions in the transmission rate and the mortality rate. So I was wondering, what about, so, and of course, the modeling approach is the SCIR model, like generally you used to do this work. Now, when someone is exposed, the length of exposure, like how long you remain in exposed, I was thinking that, well, maybe like, did you find out like whether that also is influenced by environmental conditions? Like how long you get exposed until you become infectious actually. Is that also influenced by environmental conditions in any way? Like I was curious to find out.
Yeah, I think so. As because like if you consider like beta is basically always depends on like your environmental condition, right? Like
Yeah.
from susceptible to any class, susceptible to like ACIR. Like in our case, we have like divided like from is susceptible to infected, and then exposed and asymptotic exposed actually like SEA. So there is like two compartment from S, E and A, exposed and asymptotic exposed. Like those who are like exposed means those who are officially confirmed, and those who are not confirmed, like that. So these two classes is basically very vital, as because it comes from susceptible classes, and both compartment very sensitive to the environmental condition like the beta. Yeah.
So what is the difference between the E and the I compartments?
You are talking about the infected or like
Yeah, the exposed compartment and the infected compartment. Yeah, like the difference between two.
like say for example exposed compartment, like say explain like you got the symptom, right? Like say for example for COVID, you got the symptom, so these are like exposed class. But like when you go to the hospital and you confirmed your case by testing, that is like the infected case.
Okay.
Yeah. So if you grow symptoms, that's the infected exposed class. If you go with the confirmed case to the doctor, or like you go to the your case testing, after testing, that's like infected case.
Okay. So even exposed people can still infect other people.
Exactly.
Right? Okay.
Exactly.
Okay.
Exactly.
Okay.
Yeah. So in our case, norovirus is basically very deadly, as because it depends on many cases. Like it depends on exposed, infected, as well as like asymptotic exposed as well. Like for the direct transmission, it's also depends on the environment. It also depends on the like food contamination. Yeah.
Okay.
Yeah.
Well, I don't know if I still have the chance. I have one question, and yeah, so if you can go to your slide on the transmission rate, So you had two figures on that. Yeah, one of them, you had two figures on that slide where you had BT on the left and then you fit BT to data on the right.
This one?
I have a question there that, not this one, no. No, I think it's a curve, like two figures on the slide.
This one? No, this one?
Okay, so this one, yeah, so like this one. Yeah, so what is the figure on the left? Is the left one where you have BT
Transmission rate.
it as a function of time? Is it extracted after fitting the data like the one on the other side?
Yeah, it's the same thing actually. Like say
Oh,
on the
it's
right
the
side,
same.
yeah.
Okay.
No. Like I said, like in the left hand side is basically the transmission rate, and in the right hand side it shows on the basis of transmission rate how our prediction fitted the notification data.
Oh, wonderful. Okay.
Yeah,
It's
like
fitting it
the
perfect, like it's serious. Wow.
yeah. So as because like it's mostly discretized, that's why like in transmission, like like the most of the modeling they consider beta as a like constant. So which is not appropriate like in seasonal dynamics or like if the transmission depends on other factors like temperature, humidity, or those kind of situation is transmission always varies.